The overall goal of this procedure is to observe and measure the binding of bacteria to plant surfaces.
This method can answer important questions in both plant pathology and in food safety. In particular, it can answer questions about how bacteria bind to plant surfaces and how they can be removed. The main advantage of this technique is that it's easy, quick, and inexpensive.
To prepare seedlings grown in water, place a small number of seeds in a 30, 50, 100, or 150 milliliter glass beaker. Here, tomato seeds are used. Cover the seeds with 80% ethanol and swirl briefly. Then let the seeds soak for 1 minute. Pour off the ethanol and submerge the seeds in a solution of 50% commercial bleach and 0.1% Triton X100 in tap water. Allow the seeds to soak for 20 minutes or longer if the seeds are large like bean seeds. Pour off the bleach mixture and wash the seeds 3 times with sterile water, letting the seeds soak in the water for 1 minute for each wash. Pour the seeds in a small amount of water into a sterile petri dish and incubate the seeds in the dark until the seedlings reach the desired size, between 1 centimeter and 10 centimeters. To prepare seeds or seedlings grown in sand, after sterilizing the seeds and planting materials, inoculate the seeds or seedlings with bacteria according to the text protocol. Using a sterile glass rod, make a shallow hole in the sand just deep enough to place the seed below the surface. Place the seed in the hole and cover it with a thin layer of sand. Then you seal